everyone. So today we're going to be talking about how cells allow for gene expression, and I'm actually going to be using a laptop charger to explain this. It's going to be analogous to the transcription process and the molecules and proteins that are involved in that process to allow for the expression of certain genes. So to explain what everything means, this represents the entire DNA strand. This is the gene that will actually be coded for a protein or an amino acid. And this part is the promoter, and this is the TATA box. So as you can see, the promoter is at the beginning of the actual gene expression, and the TATA box is before the promoter. Okay, so these, as you can see, are three distal control elements. And distal control elements are really important because they are parts of the DNA that aren't going to code for an amino acid that will ultimately become a polypeptide, but they are important because they serve as binding sites for different transcription proteins and transcription factors that are really important for the activation of the RNA polymerase. So here we have three, and these three distal control elements will ultimately make up the enhancer. And the enhancer is important because while multiple genes can be in different types of cells, ultimately what comes down to the expression of certain genes is the enhancer's ability to bind and connect the interaction between the promoter and other different really important proteins. So the enhancer is really important. So what happens is you have activators which come, so these will represent the activators, and they bind to the distal control elements which make up the enhancer. So they'll bind right here, and I'm just using Post-it notes. Here we go. So these activators are now bound to the enhancer, and this is important because these activators are really important because they allow for the interaction between different proteins, which will help create the initiation complex for the RNA polymerase to bind to the DNA strand. But what happens is sometimes you have repressors, which are kind of the opposite of activators, or they are counterintuitive, but sometimes useful. They will bind to either the transcription factors that are just floating around, they will bind to the activators to prevent them from binding to the enhancer, or they will just bind to the enhancer and block the activators, and this actually prevents RNA polymerase from expressing a gene. And this is important because if you have too much of a gene being expressed, it would actually waste energy and be counterintuitive for your body and its whole resource distribution. So what happens is repressors kind of do the opposite and kind of counteract the processes that normally take place in your body with the activators. So, but anyway, once the activators are bound to the enhancer, you will have a DNA protein that is responsible for kind of folding the DNA strand, and I'm using this as the protein or the molecule that will help facilitate this process, and it'll allow for the DNA strand to fold over. This is why I'm using a cord. And once it's folded, thanks to this protein, it'll be folded, and the enhancer along with the activators attached to it will be up here. The Post-its fall off, but you get the idea. They'll be attached like this, so I'll just use one for so you can understand the idea. The promoter is right here, so they're fairly close, they're folded, and what you need now are mediator proteins. So these represent the mediator proteins, and they'll bind here with the help of other proteins, transcription factors, and this will just really connect the enhancer with the activators attached to it with mediator proteins and other really important transcription proteins that are vital to allowing the contact between the promoter and the enhancer. So once this is connected, this is the final step that allows for RNA polymerase, it's just kind of chilling, it can hang out, to actually cause a transcription. So if this is all together, you have RNA polymerase, which can finally go down the strand and begin transcribing the gene, and that's how genes are expressed.